El día de hoy tenemos un invitado excepcional. Como es excepcional el título de su plática, no todos los días van a escuchar una plática que se llama Las vacunas contra la pobreza. Se dice fácil. Tengo primero que presentar formalmente al doctor Peter HOTEZ con los datos que vienen en su tarjeta de presentación. Es el decano de la Escuela Nacional de Medicina Tropical en la Universidad de Baylor. Es profesor de pediatría y de virología molecular y microbiología y es el director de la sección de medicina tropical pediátrica en el Colegio de Medicina de Baylor. Por esto no nos dice mucho, ni tampoco nos diría mucho el currículo en sí. Es un currículo verdaderamente excepcional por más de 300 artículos, la mayor parte sobre enfermedades parasitarias. Lo que lo hace verdaderamente excepcional es cómo ha logrado incursionar en el laboratorio, pero al mismo tiempo ha establecido una serie de iniciativas importantísimas dedicadas a encontrar la solución de algunos de los principales problemas infecciosos que afectan sobre todo a las poblaciones más pobres en el mundo. Él ha establecido varias iniciativas, por ejemplo, para encontrar una vacuna contra la Leishmaniasis, una vacuna contra la enfermedad de CHAGAS, y esto es lo que hace que Peter HOTEZ sea un científico absolutamente excepcional, de la mayor calidad desde el punto de vista científico, pero con una visión absolutamente inusual de cómo utilizar la moderna tecnología, la moderna ciencia para abordar los problemas de aquellos de los que frecuentemente se olvidan, de las poblaciones más pobres. Peter, es un honor para todos nosotros el tenerte aquí con nosotros. It's a real honor for Cinvestav and for all of us to have you here. You have the floor. Gracias. It's hard to do it, but it's done. Okay, it's all yours.
Thank you very much, Dr. Martinez-Palomo. It's a deep honor to be here, and I'd like to thank Dr. Asomosa Palacio for allowing me to speak here this afternoon. I want to start out with an apology, and the apology is for not giving my remarks today in Spanish, but speaking in English. And the reason I'm speaking in English was because one time I became completely traumatized by my daughter, my oldest daughter, Emmy, who once heard me lecture in Spanish. She's very good in Spanish, worked in Costa Rica, now she's getting her PhD in psychology, and she prides herself on her excellent Spanish. She heard me talk in Spanish once, and she said, "Daddy, please promise me you will never speak in Spanish again. You embarrass me terribly." She said this when she was 13 years old, where everything you say to a 13-year-old embarrasses them terribly, of course. But so I'm honoring my daughter's commitment. What I want to do today is, yes, I'm going to talk about science, but I also want to, if you remember nothing else, is to remember the potential impact that you can have as scientists, not only on science, but actually social change. And so the underlying message today is that as scientists, you are very powerful and you have the potential to have enormous impact, not only on lifting the quality of science, both here in Mexico and internationally, but actually having a very tangible impact on the lives of people, especially people who have nothing. And I'm going to dedicate this lecture to the memory of President Miguel Alemán, who I'm here this week as the Miguel Alemán Chair, and I had the opportunity to read about Miguel Alemán prior to my coming here as the president of Mexico, and the things that he did were just extraordinary, not only in politics, but also in science and health. And among his real accomplishments as president of Mexico were to help to launch the new campus for UNAM. He made fundamental changes for irrigation and farming. I thought this one was fascinating. He contained foot and mouth disease through his legislation, which I wasn't aware of. Women's right to vote, and he had a very close relationship with the United States. So what I'm going to be addressing today are this interesting group of diseases that we named in 2004, 2005 as the neglected tropical diseases. And these are a group of infectious diseases that, and you'll see this over and over again, not only occur in the setting of poverty, but are the underlying cause of poverty because of their impact on child growth and development, as well as worker productivity. And while we think of neglected tropical diseases, and that's what NTD stand for, as problems mostly of sub-Saharan Africa, increase, and when we first started, we thought about everything was Africa, Africa, Africa. We're now realizing that these neglected tropical diseases occur wherever you can find extreme poverty. And now, in the Mesoamerican region, there are at least 16 million people, roughly 1 in 10 people who live on less than $2 a day, and we've been finding these diseases widespread in this part of the world. And these are some of the new numbers that we've just collected that we're writing up now. So they include among the 160 million people in Mexico and Central America, 21 million people at risk for vivax malaria. These are studies from PAHO. 18 million children, so basically almost all of the children in the poorest parts of Mesoamerica, again Mexico and Central America, with intestinal helminth infections. Dengue fever, 12.8 million cases annually in this part of the world. Chagas disease, 1.9 million cases in Mexico and Central America. River blindness, a significant number of people at risk. 240,000 people with cysticercosis. Cutaneous Leishmaniasis, between 26,000 and 42,000. I'm happy to make these slides available if you'd like them to. Now, what that means is that every single person who lives in poverty, every single person who lives on less than $2 a day in Mexico and Central America has at least one of these infections. And the point is that these are not only affecting the health, but they're often chronic debilitating conditions. Many of these are long-standing conditions. They're actually trapping people in poverty. This is where the poverty is occurring, at least in the Mexican part. It's primarily, and this is no surprise to anybody, Chiapas, elsewhere in Mexico. But the other important point of this is poverty is not only in Mesoamerica, meaning Mexico and Central America. We have poor people in the United States, particularly in the southern part of Texas, where some of the greatest concentrations of poverty in the United States. Our new numbers indicate that we have in the U.S. 1.65 million families living on less than $2 a day in the United States. So we have profound poverty not only in the U.S. But also in Mexico. And the other reason I want to show this map is, when we think about poverty and disease coming from Texas, so much of it is said it's because it's from immigration from Mexico. But now we know this is not the case. It's not so much immigration, it's indigenous poverty in the U.S., and with it, widespread neglected tropical diseases and transmission of these diseases. So for instance, we have 1 in 10 dogs infected with Chagas disease in Texas. We have evidence of Chagas disease transmission among the homeless, dengue fever in Houston. So the point that I'm making here is that Mesoamerica does not begin in Mexico. Mesoamerica begins somewhere around Houston and then goes down. It's all part of the same ecosystem. So together with Jaime Ortega, here it's Cinvestav, Dr. Batazzi and Mary and I wrote this paper called Texas and Mexico, Sharing a Legacy of Poverty and Neglected Tropical Diseases. And if you notice in the beginning of the slide, this talk is being done jointly by myself and Dr. Batazzi. Dr. Batazzi, please raise your hand very quickly. And she is now the newly appointed Deputy Director of the Sabin Vaccine Institute, so we're celebrating this today with her. So the point is the diseases are widespread among the poor living in both the United States and Mexico. So I showed you the example of Chagas disease. These are some new numbers that we've identified in a new paper in PLOS. It's a North American disease, between 1 to 6 million cases in Mexico and up to 1 million cases in the United States as well. And to emphasize the point that these diseases both occur in the setting of poverty and cause poverty, together with Dr. Bruce Lee, now at Johns Hopkins, we just did an economic costing of Chagas disease. So Chagas disease is costing globally, mostly in the Western Hemisphere, about $7 billion and $1 billion in North America, not only because of healthcare costs, but people are too sick to go to work. And now we have this other problem of maternal to child transmission of Chagas disease. And so this brings on a new role for us as scientists, that we're being called upon as scientists not only to improve health, but we're actually improving economic development. So, so we know that of the 17 major neglected tropical diseases, which are chronic and disabling conditions, they are reducing productive capacity. So Chagas disease, $7 billion in economic losses. They are impairing intellectual and physical development in children, so we can show some of these diseases actually reduce the intelligence of children and reduce their future wage earning by as much as 40%, as well as causing adverse pregnancy outcomes. So one of the most important lessons is neglected tropical diseases both occur in the setting of poverty, and they are the cause of poverty. They are the reason why this group we call the bottom billion, the 1.2 billion people in the world who live on no money, cannot escape poverty. So this requires us as scientists to think about getting out of our comfort zone. That increasingly we have to be able to talk to people outside our field. We have to be willing to talk to economists, talk to political scientists, talk to sociologists, because what our work does has enormous implications, again, not only on health, but economic development. So that's the first point that I wanted to make. The second point is, what could we do about it? And one of the things that I think that our entire field of biological sciences is struggling with is this one. It's called the Valley of Death. And here's the Valley of Death, and here's the Valley of Death. But this is a metaphorical Valley of Death. It's a Valley of Death in product development. The Valley of Death says that we have an urgency for biotechnology as it applies in the United States and Mexico for these neglected diseases, the ones I mentioned, Vivax malaria, Leishmaniasis, Chagas disease, helminths. It also says we do have some strengths. We have strengths in basic science, we have strengths in clinical trials capacity, but a piece is missing. The piece is the Valley of Death. The piece is we do not yet effectively, for these neglected diseases, have a bridge between the basic science on one side, the clinical trials on the other side, developing an adequate bridge that will allow us to translate basic science discoveries and move them into clinical development. So what the major point I want to address today is how as scientists we can begin thinking about bridging the valley of death and at the same time not only make new products, but make new products that can improve health and also simultaneously improve economic development. So how are we doing that? Well, one of the ways that we're doing this in collaboration with Cinvestav is to work with Cinvestav in our organization, which is known as a PDP. PDP stands for Product Development Partnership. These are nonprofit organizations that use industry practices to make stuff. By stuff, I mean new diagnostics, new therapeutic products, new microbiocides and preventatives and vaccines. But the products we're going to make are for people who live in poverty and can never hope to produce them on their own. And so by these I mean new products for HIV/AIDS, for tuberculosis, for malaria, for neglected tropical diseases and diarrheal and respiratory diseases. The problem is this: until now, we've always relied on the big pharmaceutical companies, GlaxoSmithKline, Pfizer, Merck, Sanofi, to make all of the products. But what do we do for products where people can't afford them? What do we do for the 6, 7, 10, 15 million people living on less than $2 a day in Texas, Mexico, and Central America who will never be able to afford them? Who's going to make those products? Well, we don't have all the answers, but I want to tell you about this interesting group of organizations which all began around 1999, 2000. Some people call them guaranteed money-losing companies, but they're basically nonprofit organizations that are making products. An example is the International AIDS Vaccine Initiative, which is developing new HIV/AIDS vaccines, and they're based in New York, or DNDi, the Drugs for Neglected Disease Initiative, which is based in Geneva, Switzerland. Many of these are virtual organizations. They don't have their own labs, but they contract everything out. What we're doing, working with Cinvestav on, is our PDP, the Sabin Vaccine Institute, which is focusing on vaccines for neglected tropical diseases. And so we're named after Albert Sabin, who was the discoverer of the polio vaccine. And Sabin once said, and this is very appropriate for our talk today, a scientist who is also a human being cannot rest while knowledge, which might reduce suffering, rests on the shelf. And so at the Sabin Vaccine Institute, we're working to develop a new generation of what we're sometimes calling anti-poverty vaccines. Again, vaccines that'll improve health and lift people out of poverty. And here's where we're based. And I invite all of you to come and visit us because we're very close by in Houston, Texas. We're part of the Texas Medical Center based in Houston. How many people have visited the Texas Medical Center based in Houston? Raise your hand. Higher, a little higher. So a few of you have, not many. It's worth seeing because it's the world's largest medical center. It has 106,000 employees, 54 institutions, and it's basically a small city. In fact, it's really a medical city. And so what our Sabin Vaccine Institute did was to be the first PDP to move into this Texas Medical Center, in part because we wanted to be closer to the problems in Mesoamerica and work with Jaime Ortega and others at Cinvestav. And we're based in this beautiful research building here. And if you come visit us, we look like a biotechnology company, but we're a money-losing biotechnology company in the sense that we're making products for the poorest people. So our emphasis is on neglected tropical diseases, living in poverty, emphasis on the Americas, and this is the list of diseases that we're currently targeting for purposes of vaccine development. Human hookworm infection and other intestinal worm infections, schistosomiasis, Chagas disease, Leishmaniasis, and we have some new viral vaccines as well. And this is our team of scientists based in Houston. And again, we're focusing very much on bridging that valley of death to go from antigen discovery to phase 1 clinical testing. So these are the activities that we conduct. And what's unusual about it is you don't typically see these kinds of activities in a basic science research lab. They're more the kinds of activities you might see in a small pharmaceutical company where we're doing antigen discovery and structure, which is not too different from a basic science institute, but very important scale-up process development, showing we can make something the same way every time in a reproducible method to document it. Technology transfer for manufacture. We file investigational new drug filings with the regulatory bodies. In the United States, it's with the FDA. In Mexico, it's COFEPRIS. And then moving it into phase 1 clinical testing. I think the point here is that when I started as an academic scientist doing this, I would think, well, if I discover a gene or discover a protein and I want to make it into a vaccine, I just hand it off to the manufacturer, and then voila, a bottle of vaccine appears. And it turns out not to be as simple as that. There's a series of steps that you have to go through that we do uniquely at our product development partnership. And to do this, we need funding, and we get funding from all sorts of sources. We get some funding from the NIH, but we also rely on funding from the European Union, the Dutch Ministry of Foreign Affairs, the Gates Foundation. We now have some support in Mexico with the Carlos Slim Health Institute, Texas Children's Hospital, and other sources as well. So we're constantly cobbling together money from our investors, but our investors are investing not so much in because there's going to be money made at the other end. They're investing in public health and poverty reduction. So we do this, we partner with we don't do everything by ourselves. We try to partner with multinational pharmaceutical companies or developing country manufacturers in order to advance this down through the pipeline. And in this case, one of the things I'll tell you about is our partnership with Cinvestav, working with the public sector vaccine manufacturer in Mexico, which is Birmex. So here's our, again, our portfolio of guaranteed money-losing products. I'll tell you about them very briefly. I want to go through briefly our Hookworm vaccine to spend some time with. I'll talk about our CHAGAS vaccine collaboration that we have with Jaime Ortega and Cinvestav and some others. So let's talk a little bit, let's go a little bit more in details into some of the science about making anti-poverty vaccines. So this is the first one I want to tell you about. This is our vaccine for human hookworm infection. So hookworm is a highly prevalent, neglected tropical disease. Basically 50 million people in the Western Hemisphere have it. It's a leading cause of anemia, maternal and childhood anemia, in low and middle income countries. Right away, just from this alone, you can see what the scientific challenge is. This is a worm parasite about 1 centimeter long. So imagine trying to make a vaccine for something that looks like you can bite off a piece of your finger rather than something that's like a typical type of virus or intracellular parasite. This is a formidable, it's a formidable scientific challenge. It's also a formidable social challenge because it's a disease of the poorest people. Hookworm, we have some new measurements from how important it is. The new estimates suggest there's 444 million cases of hookworm infection. And we now have some disease burden measurements through an organization called the Institute for Health Metrics and Evaluation in Seattle. And at the end of 2012, they published this global burden of disease study to measure the health impact of all 291 major diseases. And they use a metric called the DALY, the Disability Adjusted Life Year. And it indicates that hookworm is responsible for about 3 million disability-adjusted life years lost annually, which makes it one of the leading neglected tropical diseases of humankind. It also occurs in Africa and India, but it's widespread in the Western Hemisphere. And here's the problem, the scientific problem that we're faced with. Here's the worm. It's about 1 centimeter in length, about ½ an inch in length. And you can see that this has pretty formidable looking teeth or cutting plates that allow it to adhere very deeply to the inside of the small intestine, which is where it lives. And here's a longitudinal picture of a hookworm deeply embedded in the mucosa and submucosas of the tissues. And then what you see is it's feeding on blood from capillaries, and then the blood is coursing out the body and then comes out the other end of the hookworm. So the hookworm is actually a cause of blood loss leading to anemia. And it's estimated that 25 of these worms will cause about 1 ml of blood loss per day. And the reason that number is important is that's the amount of blood that is equivalent to a child's daily iron intake. So a child is losing his or her daily iron intake every day because of hookworm infection. So this is a child with hookworm infection, and what I'm trying to show you is how pale she is because she's losing so much blood. And you notice she looks very puffy as well, and the reason she looks puffy, looks expanded, is because she's losing not only iron but protein with her blood. And this is causing hypoalbuminemia, a drop in oncotic pressure in the vasculature, and third spacing of fluid. And that's one part of it, but what's particularly concerning is the loss in intelligence. So the more hookworms you have, the greater your loss in IQ, the greater your loss in intelligence. And we're trying to understand what the basis of that is. So hookworms are not only stunting physical growth, they're stunting intellectual growth and reducing intelligence. So that a child with hookworm growing up in a very poor area will never get to their full intellectual potential. And now my colleagues who are economists that we work with has shown that hookworm reduces future wage earning by 40%. So these are the diseases that trap the bottom billion in poverty. And that's very important because when you're trying to convince a government to invest in something like a hookworm vaccine, it's one thing to tell them, oh, these are the diseases of the poor. It's another thing to tell them that these are the diseases that are blocking your economy from reaching its full potential. And that's a very powerful message. It's also an important disease in pregnancy. So for instance, in sub-Saharan Africa, about 7 million people in sub-Saharan, pregnant women in sub-Saharan Africa and have hookworm. And that's an important point because, as my obstetrician colleagues point out, when a woman dies in childbirth from bleeding, it's not because they're bleeding more, it's because they're starting out with profound blood loss before they ever enter into labor and delivery. So that hookworm is associated with reduced birth weight, increased perinatal child mortality, increased maternal mortality, and co-infection with malaria. So that's the health problem. Now the question is, what do you do about it? Well, there are medicines available to treat hookworms, and they include one medicine called albendazole, the other one called mebendazole. And they work very well for some intestinal worms, but for hookworm infection, the medicines do not seem to be working very well, especially when you're using them in programs of mass drug administration for a single dose. So for instance, now single-dose mebendazole seems to have low efficacy against Cátedra americanus, which is the major hookworm, with cure rates of only around 15%. And we're seeing lots of outright drug failure in some African communities. So the medicines do not seem to be working. We wonder whether there's emerging drug resistance, but nobody looks because these diseases are so underfunded. But we're seeing a number of papers in our journal, like the Public Library of Science Neglected Tropical Diseases, where we're seeing low efficacy of single-dose albendazole against hookworm. So we need to come through with a new technology. And what our proposed vaccine would be to prevent moderate and heavy hookworm infection due to the major hookworm that causes about 85% of the world's hookworm infections, Cátedra americanus, to children less than 10 years of age, and right now we're proposing it for an intramuscular injection that has 5 years of shelf life. So this is one of the first things that differentiates what we do maybe from a basic science lab, is that we set out to list a certain set of parameters that we will need in order to make our product. And what we're finding is that we work with mathematical modelers to show that whereas with the drugs, this is over time, over years, you can treat, but you're getting rebound infection over time. With the prediction is that with our vaccine, we could actually eliminate the disease as a public health problem. So let me tell you a little bit about our vaccine, because it's kind of curious. How do you make a vaccine against something that's this big, that feeds on blood? So the vaccine is currently a bivalent vaccine. It's composed of 2 antigens that I'm going to tell you about in more detail, one called GST1, the other APR1, and we formulate these antigens on alum together with a second adjuvant that I'll tell you about in a minute, using either a synthetic lipid A or a CpG oligodeoxynucleotide. So here's how the vaccine works. This is a cartoon, a schematic of a hookworm feeding on blood. And what it's attaching to the inside of the small intestine, it'll lacerate small blood vessels in the mucosa, and then the blood will course through the body of the worm, and the red blood cells come out the other end. So if you're a hookworm, you're particularly interested in feeding on the red blood cell, because that is the source of nutrients that allows you to be alive, sustain itself. So hookworms are first and foremost interested in red blood cells. And over a series of 20, 30 years, so when I first met Dr. Martinez-Palomo, I was an MD PhD student working on this problem, so it lets you know how long sometimes these problems take, working to slowly dissect out the mechanism by which hookworms feed on blood. And the steps, we think that we now have organized them into a series of steps whereby you break open a red blood cell, and so the parasite uses a hemolysin to break open the red blood cell. It then has specially adapted proteolytic enzymes to degrade hemoglobin. It's mostly interested in the globin part of hemoglobin through a series of steps, using first an aspartic protease, and then a cysteine protease and a metalloprotease, and this breaks it down to amino acids and small peptides, which get taken up by an amino acid transporter. After all of that happens, the parasite needs to do something to dispose of heme, because the heme moiety of hemoglobin is very toxic to blood-feeding parasites, because heme generates toxic oxygen radicals that can peroxidate lipid membranes, that can damage DNA. So anything that feeds on blood, whether it's a mosquito or a malaria parasite or a hookworm, has adapted special mechanisms to detoxify heme. In the case of the malaria parasite, it polymerizes the heme into an inert pigment called hemozoin using a histidine-rich protein. In the case of hookworms, they have a specially adapted glutathione S transferase that will bind up heme and transport it to different sites. So this is the mechanism which hookworms use to feed on blood. What I haven't told you yet is that this is a longitudinal cross-section of the hookworm gut, and that many of these enzymes are bound by putative GPI anchor sequences to the brush border membrane, so that as the blood is coursing down the body of the hookworm, they're being acted on by these enzymes. So what we've adapted is a strategy as follows. What if we can genetically engineer the aspartic protease over here and engineer it in a way that it folds properly, it conforms to its native structure, and then immunize an animal and ultimately a person so that it elicits antibodies? Would the animal or the human, would the hookworm feeding on blood ingest those antibodies, and then the antibodies would target to the gut to interfere with blood feeding? So in fact, that's what seems to happen. This is some work that we did in collaboration with Alex Lucas's lab, who's was with us, now on his own in Australia. And I know you're not used to looking at immunofluorescent pictures of a hookworm, but here's one right here. This is a hookworm removed from a vaccinated animal. And what we're doing is we're probing it with a secondary antibody. So it's ingested antibody to this APR1, and now the antibody is binding to the brush border membrane of the hookworm and damaging the hookworm. So, and as a result, the worm can no longer feed on blood and starves. So this is showing you blood loss in a control group of vaccinated animals, and here's the vaccinated group. So the vaccinated group is protected from hookworm anemia and blood loss, and ultimately the hookworms begin to die, and this is reduction in number of hookworm egg counts. So the problem, though, is in terms of making a vaccine, I don't think we could tell the FDA or COFEPRIS that we're going to be injecting people with a proteolytic enzyme. Why? Because it's going to damage muscles and everything else. So what do? Well, what we did was we mutated the enzyme through two canonical aspartic acid residues and turned them into alanine. So this is a cartoon of the three-dimensional structure of the aspartic protease. It looks a little bit like the continent of Australia. And what we found is that by doing that mutation, we can both preserve the three-dimensional structure of the enzyme and yet inactivate it proteolytically. And that's now made a very effective vaccine. And at the same time, what we've done is genetically engineered this enzyme, the one that's responsible for heme detoxification, to show that we can also prevent heme detoxification from the parasite, and then that dies as well. So Toya Nasojo in our group has solved the three-dimensional crystal structure of this unique glutathione transferase, and it has a very large heme binding pocket that is specifically adapted. So although it's a glutathione transferase, it has very low peroxidase activity and mostly heme binding. So we're getting up to 71% worm burden reductions when we immunize with these two proteins. And ultimately what we aspire to is to make these into a bivalent recombinant vaccine. So two different proteins, one expressed in Pichia yeast, and the other one we're expressing in tobacco plants because we've not been able to get the APR1 expressed in high yield. And then we formulate them on aluminum hydroxide in order to increase their immunogenicity together with a new generation adjuvant such as a synthetic lipid A or a CpG oligonucleotide. And so after some 30 years of work, that's to the point where we've gotten. Now the question is, how do you advance it into clinical trials? Well, the first thing you need to do, even before you start, is you want to have a roadmap for where you're going. Because after spending 30 years making these recombinant protein vaccines, we realized the science in some ways is the easy part. Because just because you've done it, now you have to show you can make it at some industrial scale, and you have to make it at low cost. So making it at low cost is key, because it's key to our what's called the Global Access Roadmap, which means ensuring that you can make that vaccine accessible to the world's poorest people. So what that means, practically speaking, is we have to be very selective on how we make the vaccine. We have to make the vaccine as cheap as we possibly can. So it means when you're using an expression vector, that you can't use a mammalian cell expression vector because it's too expensive. So we make it in Pichia yeast or in tobacco plants. It also means when we're purifying the protein, we cannot use immunoaffinity resins because that's too expensive. So we have to use ion exchange chromatography or hydrophobic affinity chromatography, and even then, the really low-cost resins. So it becomes an interesting problem of not only whether you can make it, but whether you can make it so that you can charge less than $1 a dose to do that. And then you've got to figure out a way who's going to scale it up for you. because we can make it at pilot scale in our laboratories, but how, and with Jaime, but who is going to make it for the world? Well, for the hookworm vaccine, we don't have the answer yet, but we're doing a lot of work with the Brazilians now, working both with Fiocruz as well as Instituto Butantan to see if we can ultimately make this for phase 2 and phase 3 clinical material. So there's a series of steps of how you make it, both for phase 1 material, that's the phase 1 clinical trials, which we can make at pilot scale, but how we're going to be working at phase 2 and phase 3. So the original plan was we were going to make it with the Brazilians. Then something interesting happened. We got funding to make them with the Europeans as well. So sometimes best laid plans, although we originally proposed to make it with Brazil, we got a lot of funding that came with a string attached, and that string attached was to start making them in Europe as well. So we have this new collaboration with Q Biologicals through a consortium of European and African partners known as Xxvii, which has partners in Holland, Germany, and with the idea that we'll do some of our clinical testing in Gabon with the Albert Schweitzer Hospital. And it's very exciting to be able to work with the Albert Schweitzer Hospital because he's a lifelong idol of mine. So we'll be now looking at some of the clinical development and moving it into Africa. Now, while we work on how we're going to produce phase 2 and phase 3 materials in Brazil. So currently this vaccine is in phase 1 trials. That means for safety, and so far both antigens seem to be safe in normal human volunteers. In Brazil, it also appears to be immunogenic, and then we'll be moving this into Gabon in a short period of time. Now, in parallel, we're starting to make other vaccines, including one for intestinal schistosomiasis, which is just as common as a hookworm infection is. And this one, we were able to take advantage of a completed genome of Schistosoma mansoni, a major cause of schistosomiasis, and it relies on a surface tetraspanin because it spans the membrane 4 times, and this has proven to be a highly immunogenic molecule. It's also required by the parasite for the biogenesis of the surface. So this vaccine works a little differently. It does not interfere with blood feeding, but it actually seems to help to when we use it as a vaccine, it creates an antibody response that damages the surface tegument and seems to unmask the immunological properties of the schistosome. So this is also now as a monovalent vaccine with this antigen, just tetraspanin surface antigen known as TSP2, formulated on alum. And so we've just now completed the manufacturing of this, and now we're filing the investigational new drug application to begin phase 1 clinical testing. We're also looking at the possibility of whether we can look at folding in other antigens into our scaffold of the hookworm vaccine to target multiple worms at the same time. So one of the holy grails we're working on is the possibility of advancing a multivalent pan and helminthic vaccine. Could we target most of the major worms affecting children in resource-poor countries by adding Ascaris antigens or Trichuris antigens at the same time? And that's something we're quite interested in doing. Now let's move to a totally different type of problem. And it's a totally different type of problem because it involves a completely different arm of the immune response. And this is how we might go about making a vaccine for Chagas disease. Chagas disease, as you know, is caused by Trypanosoma cruzi. It's an intracellular parasite. And we've been working with a collaborator in Mexico named Eric Dumontil, who's discovered 2 very promising antigens known as TC24 and TSA1. And he discovered this using DNA vaccines. The problem being with DNA vaccines is, and the reason he's using a DNA vaccine is, whereas for the hookworm vaccine and for the schistosomiasis vaccine, we're trying to achieve high levels of antibody that neutralizes this enzyme. For both schistosomiasis and Leishmaniasis, it's a different problem. We require high levels of cytotoxic T lymphocytes and Th1 immunity. So the first question becomes, can we do this? Can we achieve this with recombinant protein antigens? Well, the first is see if we could scale up the antigens as recombinant proteins. And we've had a lot of success in our laboratory for this one, TC24. With TSA1, we've been working very closely with Dr. Ortega's group, has had some success now at refolding this from E. coli. Now the question is, can we stimulate a Th1 immune response? It looks like we can, but we have to use a lot of novel try a lot of different novel adjuvants in order to make that happen. And one that's looking very exciting is a synthetic lipid A, known as glucopyranonyl lipid A, that's formulated in a stable emulsion. So we're doing preclinical testing now in Houston and Mexico, and we're hoping through our consortium of partners, which is based at Sabin and also includes Cinvestav, whether we can advance this into the clinic. In this case, with the manufacturer being Birmex, the public sector vaccine manufacturer here in Mexico City. Now, we're also concerned, though, that we may have to try novel approaches in order to stimulate Th1 immunity. And one strategy that we're looking at is whether putting these antigens into nanoparticles might be a way to achieve strong Th1 immune responses. And working with Kathy Yao at our institution, using atomic force microscopy, she's able to now visualize the TSA1 antigen that Jaime is working on in his laboratory to show that it can appear on the surface of the particle. Now, the other question is whether this will ever get out of the laboratory and into the clinic. We know that the FDA and organizations like COFEPRIS in Mexico or Anvisa in Brazil has a lot of experience with the recombinant protein antigens, but particle antigens on nanoparticles is something completely different. So even though it's exciting science, it may slow us down from a regulatory perspective and make that more difficult. So we have to weigh all of these business decisions into making vaccines for the poorest people. We have a cutaneous leishmaniasis vaccine. I know I'm coming close to the end, and I want to finish up on that. We're also working with Jaime and others on. We're finding, curiously, a lot of cutaneous leishmaniasis in Texas, and a lot of them are from Cuban immigrants who are getting visas to go to Ecuador and Colombia, and then they're being brought up through the jungles of Panama and then declaring asylum in Houston. So we've seen large numbers of cutaneous leishmaniasis cases in Houston, which is quite ironic. And so we're looking at a bivalent recombinant protein vaccine strategy that uses a Leishmania antigen, but we're also collaborating with Jesus Valenzuela's group at the National Institutes of Health, who's identified a number of sandfly proteins that have very strong adjuvant properties for Th1, because again, we appear to need high levels of cytotoxic T lymphocyte activities for. And so some of these sandfly proteins have quite an interesting structure. So Toya Nasojo in our group has solved the crystal structure of some of these sandfly antigens, and you can see that they're very rich in alpha helix. And we're wondering, is all that alpha helix anything to do with the Th1 stimulating properties? It's something we don't know about. We have a new SARS vaccine as well, which I know is not a neglected tropical disease. It's more of a biodefense problem. This one is looking at the receptor binding domain of the SARS vaccine, and where we seem to be achieving high levels of neutralizing antibody with that. So right now our status is we've got a hookworm vaccine in clinical trials, we have a schistosomiasis vaccine in clinical trials, and my hope is the next time we come to talk to you, we'll be able to talk about the ones we're doing in collaboration with Cinvestav for Chagas and Leishmaniasis, and get them moving into clinical trials as well. And this is how I'll end. which is to say, I talked to you a lot about the idea of using a vaccine to lift people out of poverty. I also think the social good of vaccines could even go one step further. And to explain that, I just need to tell you the story, if you'll bear with me, about a story that not many people know, how Albert Sabin developed the oral polio vaccine that most of us took as kids. And so the virus strains for the oral polio vaccine were developed in Dr. Sabin's laboratory in the United States, and this was happening around 1958, 1959, and 1960. Well, at that time, the Soviet Union, the USSR, was experiencing horrific polio epidemics across the USSR, and they were very interested in acquiring Dr. Sabin's technology for making the oral polio vaccines. So not many people know it, but this guy here, Dr. Chumakov, who's a Soviet virologist, got back-channel permission from his bosses to come to the United States, and Sabin got permission from his bosses from the U.S. State Department to work with Dr. Chumakov. This was happening in 1958, 1959, 1960, at the very height of the Cold War. Two countries were able to put aside their ideologies and collaborate together to make the vaccine. So not many people are aware of it, but the oral polio vaccine was actually developed, even though the strains were discovered in Dr. Sabin's laboratory, it was developed in a vaccine in the Soviet Union in 1958, 1959. This is a telegram we got a copy of from Dr. Chumakov to Dr. Sabin saying, "Heartily grateful for sending your polio strains. Received everything September 25th on dry ice. Start to work in November. Very yours truly, Dr. Chumakov." The vaccine was tested on tens of millions of Soviet school children, shown to be safe, and those were the pivotal studies that led to licensure. So the question we're asking is, there's a great opportunity, I think, in joint vaccine development to build international relations. Sure, between the U.S. and Mexico and Brazil, but what about countries that the United States does not always agree with ideologically? What about the opportunity to make vaccines with the Venezuelans or the Cubans or the Iranians? Is there an opportunity to take that a step further? And so this is something that we're looking into. There are a lot of political barriers to overcome, but what do you do when you get a telegram, not a telegram, but an email, the modern day telegram from an Iranian scientist, as we have, say, look, we need a Leishmaniasis vaccine. You guys are working on a Leishmaniasis vaccine. How do we work together? Well, here are some of the problems. One, what do you need to make a vaccine? Well, I was telling you we need fermenters, we need large-scale protein purification equipment, and we need PCR vaccines. The problem from our State Department is, what do you need to make a bioweapon? Large-scale fermenters, large-scale protein purification equipment, and a PCR machine. So anything we would do with a vaccine is potentially dual use. How do we get around these kinds of laws in order to collaborate together for making a vaccine? So it's a way, again, that as scientists, you could be called upon not only to work with the economists, but you may have to work with people in your government or Department of State or the equivalent to explain to them the importance of this concept of vaccine diplomacy. And with that, I'll thank the leadership again from Cinvestav and the Miguel Alemán Foundation for allowing me to come to address you today. It was a great honor and privilege to talk to you. Thank you.
Muchísimas gracias por este viaje que nos ha llevado a Peter HOTEZ sobre el complejo mundo del desarrollo de vacunas para enfermedades olvidadas. Como es tan generoso, él es conocido por muchas cosas, entre otras por su generosidad. Nos va a dedicar unos minutos para contestar algunas preguntas o algunos comentarios que quisieran hacer ustedes. Así es que está abierto a preguntas. El doctor Meraz quiere hacer una. ¿No hay micrófono?
I got the phone here, the microphone here. Is it all right, Marco, if I start, or would you like to go further? Okay. Dr. HOTEZ, here. I'm very pleased that the Miguel Valeman Foundation was able to support this visit, and we're very happy to have you here at Cinvestav. And as you know, we invited you for ICOPA, so we'll hope to see you again in August. And I've got two questions. One is, there are two very important aspects on vaccination, and maybe more, but those two that I refer to is the cost of vaccines, one, and the other one is how vaccine actually helps in terms of being long-lasting as opposed to short-lasting, which means also that will cost a little bit more, well, but probably cost more. So what are the prospects for all those really good vaccines that you just talked about?
Yes, thank you for that question very much. So the questions are about cost and length of protection, and it turns out that the two are linked in a very profound way. So in terms of costs, we work with economists who do economic modeling of our vaccine. And to be competitive and to make a product that will actually be used, we have to make it for $1 a dose. If we make it, roughly, I mean, maybe as much as $2, preferably closer to 50 cents. So how do you make a vaccine at that low cost? So that's why, as I mentioned, we tried to focus on very low-cost expression vectors. E. coli, the old-fashioned ones, E. coli, pichia, yeast, in some cases tobacco plants. Because if we start using mammalian cell expression and making a $20 vaccine, it's nice, but it'll never be used. So we have to make it under a couple of dollars. And that gets us to length of protection. Well, we look to the advice of the economists who tell us that it should last at least 5 to 7 years. So we've got to make a recombinant protein vaccine that lasts for 5 to 7 years. Otherwise, it's not going to be used. So the bar is high, but that's what we're working on trying to achieve.
Peter,
yes.
Thank you very much for this wonderful talk. I'm surprised how many things you have achieved since we were students. I was students with Isaura Mesa, and
sure,
yeah,
I remember, of course, yes.
And something that surprised me is how you can achieve this intellectual concentration of immunologists, biotechnologists, because otherwise it's almost impossible to get into all these strategies that you has been developed in order to, attack the metabolism of a particular organism, or the structure, or how it's going to be possible that all these technologies achieve this low cost and be spread along the population, because that's going to be the big challenge in order to produce this
right.
amount.
So there's a couple of, it's a great question, and it's actually a very profound question, because people at a very high global policy level are dealing with this now. Not so much with us, but with the pharmaceutical companies. So the concept is called, and there's a big battle right now going on in Geneva and places like that, and the battle is over something called delinkage. It means that the reason that GlaxoSmithKline or Merck are charging $150 for Prevnar, the pneumococcal vaccine, or they're charging more than $400 for the human papillomavirus vaccine, the cervical cancer vaccine, is not that it costs that much. I mean, it costs more than $1 a day like our vaccines, but it's not costing $400. What's happening is the pharmaceutical companies need to recover their R&D costs, and so they fold it into the cost of the vaccine. So what people are screaming about is, look, if you're ever going to make low-cost vaccines for people in poor countries, you've got to delink it. In other words, what we're going to have to do is, I don't know if I can show you here again, is take our donor support and tell them, look, we're not going to be able to pass on all your generous support that you've given us onto poor people. We've got to figure out a way to all of these costs from places like Hookworm And the other donors that I told you about, we've now got to figure out a way. We're going to have to absorb the costs ourselves and not pass that on. So that's, there's no other way. There's no other way it's going to happen. So, but you know, the drug companies are in the business, not that they're bad guys, but they have to make money for their shareholders. So the idea is all of our donors have bought into the fact that they're never going to see return on their investment in terms of dollars. The only return they'll see on their investment in terms of public health and poverty reduction. I hope that kind of addresses it.
I'll also, sorry, I'm here.
Good.
Hello. So first, thank you again and very welcome to Mexico. I have two questions on the two spectrums. I'll start from where you just ended. And the question is, how does the drug industry treat the PDPs? A comment on that.
It's a great question. I think when PDP started, the drug companies didn't know what to do with us. I think now what we're seeing is greater interest in partnering. And so that's kind of nice to see. So we've had discussions with Pfizer, GlaxoSmithKline. Maybe we can partner in certain limited ways. And part of the reason is that, you know, the drug companies, you know, I once heard a very interesting talk from Jean-Paul Garnier, who at that time was the CEO of GlaxoSmithKline. And he said something very interesting. He said, you know, my company has 100,000 employees. I cannot motivate 100,000 employees to come to work every day just to make money for my shareholders. Our employees need to know that we're doing something good for the world. So the companies themselves realize that to motivate their employees, they need to be taking on some of these types of projects. And they have, but starting slowly. I mean, we've gotten inquiries from most of the major pharmaceutical companies. We haven't quite figured out a way to partner in a meaningful way yet. Other PDPs have. So for instance, DNDi has been very successful in this. So I'm hoping that will happen over the next few years. It's going to happen, I think.
If nobody else, then if you will allow me to take you now to the other end of your spectrum, your science.
Sure.
I don't understand how the glutathione S transferase can take care of heme. If you can enlighten me.
Well, what it does is, what we have found is that the enzyme has a very low peroxidase activity. And it appears to be the nematodes, I'm trying to get to the picture here, it's a little slow. The nematodes that feed on blood, which include hookworms, but also some veterinary helminths as well, have a specially adapted glutathione S transferase that has very high binding constant for heme, something that's way beyond any other kind of GST. So it seems to be mostly functioning in heme binding. So we're hypothesizing that and so that's observation number one. Observation number two is that when we elicit, it works as a very good vaccine. And now we have to link the two. So the third observation is that we find that we can elicit, when we immunize with this, we can elicit antibodies that neutralize the heme binding function. Actually, the antibodies will block heme. So we've tied that all together to make a hypothesis that the way this vaccine for GST works is by interfering with heme binding and then allowing free heme to go throughout the nematode and cause destruction.
Okay, but then the issue is you would either need to have a lot of GST1 to deal with all the heme of the blood, or there needs to be a subsequent step where heme gets deneutralized, passed on to a heme oxygenase or something to release the iron. But thank you.
Right. So, you know, this is our working hypothesis, and it seems to be backed up by data. Now, what happens after heme binding? I think that's a very important question. It's being transported. Is the parasite using the iron from the heme to do something? We don't know that yet. One of the problems that we face working with something like hookworms is that there's no in vitro culture methods. Genetics is very difficult. We've tried and failed to get RNAi to work. So RNAi works for C. elegans, but it hasn't so worked for hookworms. So I like to tell people, other than the fact that you can't get enough material to do biochemistry, you can't do genetics on, and you can't do cell biology, it's a great organism to work with in the laboratory.
Thank you.
Great, thank you again for coming.
Pues evidentemente estamos todos muy agradecidos con el doctor HOTEZ por esa magnífica conferencia. Yo quisiera hacer algo inusual. Parte de nuestra cultura nos reúne aquí en la ciencia, pero nuestra cultura también está en la literatura. Y yo quisiera pedir un momento de silencio para recordar a José Emilio Pacheco. Muchas gracias. Creo que hay pocas dudas en ninguno de nosotros que era el escritor mexicano más valioso que teníamos. Gracias.